All right, everyone. We're going to go ahead and get started. Thank you so much for being here. It's great when we get to all get together and be in community. So I thank you so much for being here. This is our research seminar, and it is a hybrid event. So that means we have folks online and folks in the room. If any of you haven't done one of these with us before, we just ask that if you're online, go ahead and put any questions you might have into the chat box. And we have a volunteer here, Roseanne. Thanks, Roseanne, who will make sure to help you with any technical issues or read out your questions for today's speaker. For folks in the room, if you have a question for today's speaker, when we get to the question and answer section, if you just raise your hand, I will bring you a mic, and that way folks online can participate with us as well, even if they can't be here at Hatfield. But again, I just want to say thank you so much for being here and showing support for today's speaker. I have a quick couple of announcements, and then I'm going to get started here. I just wanted to promote next week's seminar, February 13th, and the speaker, I believe, is in the room. I think I saw that Doing Song is a research associate with Coombs. Will be here talking about RNA sequencing and the regeneration capacity of hydractinia. And so that one is going to be great, and we'll get to learn a little bit more about work that they have been working on before they came here to Hatfield. So I'm excited to learn a little bit more. I wanted to let everybody know that we do record these seminars. So if you are interested in any of our past seminars or sharing today's seminar, just go to the HMSC website, go to the events page, and you can find the seminar series there. You're also able to find a calendar for upcoming events. But I just want to jump into today because I'm excited. So I want to introduce Dr. Emily Slesinger, who is a research biologist with NOAA's Alaska Fisheries Science Center here at Hatfield. She attained her bachelor's in marine biology, her BA in environmental science at UC Santa Cruz, and her PhD in biological oceanography from Rutgers University. She has a background in fish psychology. I like that. Physiology and reproductive biology and fisheries science. And her research tries to integrate all of these disciplines, including psychology, in investigating environmental stressors into fisheries populations. Her current work examines how ocean warming, ocean acidification, and ocean oxygen affects commercially important Alaska fisheries species. When she's not nerding out in the lab or at her computer, you can find Emily either outside on trail running, gravel biking, camping, fishing, and she just wanted to let everybody know her favorite fish is the Pacific spiny lumpsucker because they're just adorable. And if you haven't seen one before, they'll be on display in our visitor center in a couple of weeks because we got some little babies in the back. But I would like to hand this mic off to the cod mother, so we get to learn a little bit more about what's going on. Thank you very much.
Okay, everyone can hear me okay? Perfect. Well, thank you everyone for coming in person and everyone online. I'm really excited to share with you all what essentially is a really large experiment we wrapped up last year. And before I get started, I'm actually just going to do my acknowledgements now. I want to thank all of my co-authors listed here, as well as our funders, the Ocean Acidification Program, for funding this research. And of course, I would be remiss not to share with you all the Pacific spiny lumpsucker, if you don't know who they are. They are a tiny little ball of a fish, but their pelvic fins are modified suction cups, which means that they can onto things, and that's kind of what they do for the majority of their life. And I think that they're really cute, and I love them a lot. But alas, today is not going to be about the Pacific spiny lumpsucker. Today, our focus will be on Arctic cod. And we will go over the effects of ocean acidification as advertised, but we're also going to weave in this story about fish reproduction. And even if you know a lot about fish reproduction or a little bit about fish reproduction, I hope that everyone can come away today learning something new and sort of instilling this curiosity, awe, and wonder for the biological world, something that I think is always important, but maybe especially so nowadays. So, as you might have heard in my bio, I kind of don't fit in one group of people. I kind of wear many hats, and that tends to fall into the camps of fishery science and fisheries biology. And I think a lot of us probably are also quite interdisciplinary and maybe feel that way as well. So in my world, of course, the main goal is fisheries management. And this is going to be like setting things like the timing of when you can go fishing, maybe how many fish can be caught, or if the locations of where you can catch fish differs. And in order to inform these types of regulations, you need to know a lot about the population dynamics. You need to know about how that population is growing or decreasing over time. Maybe you need to know about genetic substocks or some substructure in the population. And then you also need to know something about how many fish there are in certain size classes. And that's particularly important if you have a species where you are setting a minimum size limit for how many can be caught in a given year. I really like to wear the hat also of a physiologist. And of course, the term physiology is incredibly broad. But what I like to typically focus on are questions related to energy dynamics, lipid biochemistry. And I apologize, I couldn't think of anything better than butter, but also like to focus on metabolism, metabolic rate, energy production, and then also hormones, typically as they relate to either reproduction, growth, or some capacity of immune system responses. And importantly, environmental stressors have the capacity to affect everything you see on this screen. So this could include things like changing temperature, could be ocean acidification or changes of the CO2 concentration in the water. And it could also be hypoxia, the decrease in oxygen content in the ocean. And in my opinion, I think reproduction is this beautiful umbrella that helps tie together all of these potentially disparate subdisciplines that we have in fish biology and fishery science. So just as an example, say we were focusing on the effects of temperature and say we knew that there was some effect on the physiology. Maybe there was an intense increase in metabolic demand. That might lead to a negative effect on reproduction. Maybe they just can't produce as many babies. That ultimately will affect the population dynamics. Maybe the recruitment for that year is a lot lower. There's not as many young fish that make it to the system. And if we know that, theoretically, we could help inform fisheries management. And of course, this is an incredibly idealized example, and nothing in life works this simply, but I think it illustrates that reproduction really can be this nice tie together of all of these different sciences that we like to focus on. And that brings me to the wonderful world of fish reproduction. So almost anything that you could think of, a fish can probably do it. So the most common mode of spawning in fishes is broadcast spawning. So that's releasing male and female gametes into the water column. And of course, at the opposite end of that spectrum would be internal fertilization, which is what our elasmobranchs do. And fun fact, they are the ones that invented that. Fishes can also do internal fertilization. This is an example of a rockfish ovary, and they will develop those eggs inside of the ovary and hatch out live larvae. So all those little sparkly dots you see are the eyeballs of the larvae. Another idea of maybe protecting those vulnerable eggs before they're ready to hatch would be mouth brooding. So in this case, many times eggs are laid typically in the male mouth, and then they keep those eggs nice and cozy until they're ready to hatch. We can also have incredible complexity in spawning, right? Salmon are a great example of living an entire life in the ocean and then needing to find their natal river, setting up nests and spawning, and then dying. And then sex change is incredibly common in fish biology. We can have simultaneous hermaphrodites, we can have sequential hermaphrodites. In the sequential hermaphroditism world, we have protogeny, so that is female to male, and protandry, male to female. And these are California sheep's head. They're protogynous, so the male in the far left was once a female, and typically this is a display that they do to maintain territory. So now I want you to think about your favorite fish. And if you're thinking, Emily, I don't have a favorite fish, then maybe think about a fish you like to eat, or maybe one that you've seen in an aquarium, or it could be the Pacific spiny lumpsucker. And I want you to think about whether or not you know anything about its reproduction. And even if you don't, try to start thinking about what sort of questions would I have to ask myself if I put myself in the fins of that fish of how to be reproductively successful. So for example, a question that you're going to have to ask yourself is, how many times can I reproduce? Right? Am I one shot, one opportunity like a salmon? Or can I spawn multiple times over my lifetime like a cod? Another question you might have to ask yourself is, how many offspring do I produce? Do I put in a lot of effort into a couple of offspring that look really good? Or do I don't put a lot of effort in my offspring, but I make millions and millions of babies with the idea that some of them will be successful? In reality, there are tons of questions that you could ask yourself about how you might be reproductively successful for your specific spawning strategy. And one that I like to highlight in regards to how much reproduction is important is that reproductive maturity typically can drive growth trends. So if we were to look at some organisms looking at how they grow over their lifetime, you might see an individual like this person who grew, and then reached a maximal terminal size in like 4th grade. And then I don't grow ever again. And while I might shrink, that's not from like a growth dynamic, it's more deterioration. So that's determinate growth. That's in contrast to, say, something like a shark. Most fishes have indeterminate growth. And so essentially what that means is throughout their life, they will grow. Early in life, they will grow. And at some point, there will be some deceleration of growth. And then they will continue to grow throughout their life, but at very, very small increments. And we can see that for small species too, like a goby. They're not going to live as long, they're not going to get as big, but they will have this sort of the same trend. And what's important is that division of where we see that deceleration in growth is typically when those species are, or the individuals are reaching reproductive maturity. And also for humans. I did not become mature in 4th grade though, so that doesn't make sense. But anyways, but essentially what it means though is that at some point, if you're going to finally be able to reproduce, there's so much energy that you need to shunt into towards reproduction that's now going to take away from the energy that you have for growth. So if we go back to our question cloud, another grouping that I like to think about a lot when it relates to spawning strategies is this question about time. When do I reproduce? How long do I have in the year to reproduce? Can I eat where I'm reproducing as well? And I'm sure maybe the whale people thought about that question. So if we think about a given calendar year and we were to look at 2 examples of the extremes in high and low latitudes, the first big difference here is going to be the seasonality, right? High latitudes are driven by really intense seasonality, maybe by temperature, sea ice, light, or a combination of those. And that's in contrast to the low latitudes with a much more dampened seasonal signal. What's important about that seasonality is it can set the stage for when is a good time for the larval or the baby animals to eat. So in high latitudes, that's typically in the summer. And not only do they need to eat when there's abundant food, but they also need to grow as fast as possible before their first winter. And that's in contrast to lower latitude systems where maybe the optimal feeding time is kind of constant throughout a good portion of the year. And so with that sets sort of the stage about when adults might spawn. So in the high latitude example, a Pacific cod is going to spawn in the winter, and that's going to maximize the amount of time that those larvae have to eat until their first winter. And not only are they going to try to spawn in the winter, but they're going to use what's known as a total spawning strategy. So this means that they develop all of their eggs at once and they release all of them in one go. And that could be in contrast to, say, a red snapper. They can maybe also spawn sometime in the winter, but they're going to release a couple eggs, but they're going to do that throughout the year. And that's known as batch spawning. And so that difference in spawning can also set the stage for the feeding times for those adult animals. So in the high latitude example, typically we have what are known as capital breeders. So they're going to eat outside of when they're spawning. Essentially, when they're spawning, they're not eating because either there's not a lot of good food there, it's not the right time, or in the fish's case, in that total spawning strategy, their ovaries get so big that there's just not enough room to have also a big stomach. So that can also be a limit on eating. And that's in contrast again to that low latitude example where they're more of an income breeding strategy. The food that they're eating is going to typically be directed towards reproduction throughout the year. And what I hope to illustrate with all this is that reproduction is essentially a game of trade-offs. And The reason being is that reproduction is costly, but we haven't really gone into what makes it so costly. And so this requires a little deep dive into physiology, and it's in the reference of females. But essentially, most fish store energy in the liver. The liver is also an important site. It makes and stores vitellogenin, a lipoprotein, and vitellogenin is directly incorporated into developing eggs inside of the ovary. That vitellogenin, when it's inside of those eggs, is going to be useful for embryogenesis, or sorry, not yet. Then they have the egg, and that vitellogenin is important for embryogenesis as an energy source during that time point, but also it can also make up some of the yolk for the hatched larvae. And so essentially what this means is we have a direct energy shunt from the female's energy stores to her reproductive output. And it can be so intense that you can measure that drawdown of energy during a spawning season just by liver weight. So it's pretty costly. And because it's costly, it's likely that environmental stressors also have an effect on reproduction. And we can kind of view this in two different lenses. So the first would be in the lens of energy allocation, which I kind of keep alluding to. But essentially the idea is that there's a finite amount of energy for that fish. Most of it, or a good chunk, will go to basal maintenance or metabolism, kind of keeping the internal machinery running. And then the rest is going to go towards things like reproduction, activity, growth, and storage. And then say there is some exposure to an environmental stressor, that metabolism might increase substantially, and that's going to leave less available energy for things like reproduction. The other lens that you could view this through is more from that of endocrinology and sensory biology, but essentially environmental stressors can act on the fish body at multiple parts, including the brain, pituitary, and gonad. And without going into the weeds of endocrinology, essentially there are very complicated feedbacks between sex hormones and glands and the brain. And so environmental stressors here can sometimes affect things like the timing of when spawning is going to happen, maybe the amount of energy that's going to be put towards spawning. And so it can act in sort of these two different ways. For today, we'll be focusing on ocean acidification. And I'm sure everyone has seen the Keeling curve, but there's two things that I want to point out. The first of which is atmospheric CO2 has been increasing and oceanic CO2 matches that. So that's what the green is. As that increases, we also see the decrease in oceanic pH or ocean acidification. For fishes, we're concerned about that ocean pH component, but also the increase in CO2 concentration. And in fact, for the rest of this talk, when we talk about ocean acidification, it'll actually be in reference to CO2 level and not pH. In the Arctic, we have been seeing the effects of ocean acidification, both at the surface and at the bottom. And depending on what climate change or climate scenario you're looking at, basically, there can be a high degree or a pretty big degree of continued ocean acidification into the future. And within the Arctic, we are concerned about polar cod or Arctic cod, depending on where you are in the Arctic. They are an incredibly important forage fish species, and they help shunt energy from lower trophic levels to the higher trophic levels. That includes things like marine mammals, seabirds, and other predatory fishes. Arctic cod are circumpolar, so you can find them throughout the Arctic, and we primarily focus on the Alaskan Arctic population. So importantly, within that region circled in yellow, whether or not you include ocean acidification can change the future predicted abundance of Arctic cod. But these authors pointed out an important component, which is we have a high degree of uncertainty. So that really deep purple is pretty high uncertainty, and that's especially prevalent in that Alaskan Arctic region. And that uncertainty just comes from we just don't know enough yet. So that's where we need more laboratory studies to figure out how ocean acidification affects Arctic cod. So we've been focusing on ocean acidification and Arctic cod in a couple of different experiments. And this one in particular is focused on the effects on reproduction. And in order to do it holistically, we wanted to start exposing female Arctic cod at the beginning of when they start developing their eggs all the way to larval hatching. So for Arctic cod, that's a 5-month-long experiment. So back in October of 2023, we started exposing Arctic cod to either ambient or control conditions or high CO2 or ocean acidification conditions. Sorry, I'm also getting over a cold, so I'm trying not to have a coughing attack. Okay. Good. In February, the fish are ready to spawn, and we're able to strip spawn them, which means that we can take out the gametes from the males and females and keep track of the parents. We fertilized gametes from adults from the same tanks, so the same ocean acidification treatment. And we also sampled the eggs at this time point for lipids, so that's prior to fertilization. We then placed those fertilized gametes into egg incubation beakers, again at the same ocean acidification treatment. And Arctic cod take about a month until they're ready to hatch. So daily, we would go in and count and remove dead eggs to get a mortality schedule. And then in March, Arctic cod are ready to hatch. And this happens over a prolonged hatch cycle, so across many days. So once they start hatching, we go in and count and remove hatchers daily to get this nice time series. It also allows us to get an accurate hatch success. And then we also photograph them so we can measure some morphometrics later. And we'll get into that a little bit later in the presentation. Okay. So with this type of data, there are sort of two parts that we could divide it into in terms of analysis. The first is mostly focused on those spawning adults. And that's going to ask the broad question of what drives these differences that we might see in the adults. And in part two, we can look at those maternal effects. As I said before, we're able to keep track of the offspring from the moms. And so here we can ask the question of, do differences in female fish affect offspring survival and condition? And so we'll get started with the spawning adults first. We kind of had four broad categories of things we looked at. We looked at energetic condition, reproductive output, egg morphometrics, and egg quality. And when we were analyzing this data, we always assessed whether or not there was an effect of CO2 level or ocean acidification. We also assessed whether or not there were tank effects. So this would include things like size and sex ratio, and that's because we didn't know the sexes of the fish before we put them in. And then we also looked at tank temperature. And what I'll say briefly is our tank temperature differential was between 0.1 and 0.3 degrees daily. And that does add up slightly over time, so we just wanted to make sure whether or not there was an effect from temperature. And while we go through the results, I'm going to show you essentially the best fit model of a bunch of things I did behind the curtains. I'm not going to get into the stats today, but if anyone has any questions, I'm happy to go over that later. All right, so we're going to start off with energetic condition. And here we just have one metric. We're looking at the hepatosomatic index. This is the ratio of liver weight to body weight. And a higher hepatosomatic index would indicate higher energetic condition. So right off the bat, there was actually no effect of anything on male hepatosomatic index, but we did see an effect on the females. And here we actually find that tank temperature mattered. We see that for females in warmer tanks, they ended spawning in lower energetic status than the females in the colder tanks, and this suggests that there is an effect of that tank temperature differential. Next, we're going to move on to reproductive output, and here we have three different metrics to look at. We have the gonatosomatic index, which is kind of the sister to hepatosomatic, but instead of liver, we're looking at gonad. We don't have this for the females, and that's because we strip spawn them, so by the time we dissect them, we don't have a gonad weight. But we can look at the fecundity or the number of eggs that came out of the ovary. Arctic cod, like those Pacific cod, are total spawners. So they're going to make all of their eggs at once, and they're going to be ready to release all of them at once. And then for the males, we also looked at spermatocrit proportion, which essentially is taking the milk, spinning it down, and seeing how much of it is packed sperm in reference to how much of it is seminal fluid. And so we'll actually start off with the spermatocrit proportion first. And we actually see that there's an effect of male size on the spermatocrit proportion. We see that the larger males had lower spermatocrit proportion than the smaller males. And this is actually kind of interesting because it could suggest that there is some sex competition that occurs. When this has been seen in other fish species, the idea is that these big males are competitively advantageous. So the small males instead put energy into sperm production in the hopes that during spawning events, they can kind of sneak in, crop dust as much as they can and get out before someone notices. We don't know a lot about Arctic cod spawning underneath the ice, so it's just a theory, but interesting to see. Next, we looked at the gonadospermatic index of the males, and here we actually see an effect of CO2 level and the hepatosomatic index, or their energetic condition. The males that were in higher energetic condition also had higher reproductive output, which may suggest that there is an energy demand for reproduction in the males. Usually we don't think of it as highly energetically taxing. We also see an effect of CO2 level where there's a decrease in reproductive output for the males that were in ocean acidification conditions, which could be a negative effect of ocean acidification. And while I said that we don't typically think of males having a big energetic draw for reproduction, if anybody knows gonadospermatic index, these numbers are huge. So usually for males, the numbers are closer to like 1 to 6 during spawning. And here we have these really big numbers. So again, there might be some interesting things going on with the males. We'd have to explore it a little bit more. And then finally, for the females, looking at fecundity, we see an effect of the female size and the CO2 level. Here we see that bigger females have higher fecundity, and this is typically something we expect to see. But we also see a positive effect of ocean acidification. Their fecundity is increased under ocean acidification conditions. We have actually seen this in other fish species as well, but this might be the most high latitude example that I can think of. Okay, so now we're going to move on to the egg morphometrics, and we're done talking about the males. So for the egg morphometrics, we looked at 3 metrics: diameter, size, dry weight, so that's taking out all the water. And then we looked at egg concentration. And essentially, egg concentration is, if you could imagine squeezing the contents of an ovary into a beaker, there are going to be eggs, but there's also going to be some ovarian fluid. And we actually found variation in the females, some that had more or less eggs for the same amount of ovarian fluid. So going through egg morphometrics, we found no effects. We did find that there were correlations within the egg morphometric groupings themselves, but there was no effect of CO2 level, energetic condition, or any of those tank effects, but they are important, so there is a reason we went over them. And then finally, looking at egg quality. Here, these metrics are related to lipids. So the first is lipid concentration. This is the total amount of lipids in an egg, and then we can break out those lipids into lipid classes. And so the 3 major lipid classes in eggs are polar lipids and sterols. Those are kind of structural membrane-associated lipids. And then we have triacylglycerols, which are more energy storage lipids. And then there is a small amount of free fatty acids, and that's essentially a fatty acid that was attached to a carbon backbone and is no longer. And then we can also look at a tag to sterol ratio, and that's kind of like an egg energy condition index, essentially comparing how much energy is available for usage versus what's available, the lipids that are available for structure, which is important to build that body during embryogenesis. So starting off with lipid concentration, here we actually see an effect from those egg morphometrics. We see that there's an inverse relationship between egg lipid content and egg concentration. So the females that had lower egg concentration, so fewer eggs for the same amount of seminal, ovarian fluid, have a higher lipid content in the egg. And this could suggest that there's a trade-off between how many eggs the female is producing and the quality of her eggs. Next, we're going to look at free fatty acids, and you're going to be like, wait, you said that doesn't really matter. Why would we go over it? And that's because we found this interesting response with CO2 level. We found that for the females in ocean acidification conditions had higher levels of free fatty acids in the eggs that they made in the ovary. And what's interesting about that is these eggs are not alive yet. This is directly coming from the mom. And so it suggests that there might be some lipid breakdown occurring during oogenesis. And that could be a negative effect of ocean acidification. And then finally, looking at the tag to sterol ratio, again, that's kind of like this egg energy condition index. And we actually see an effect of tank temperature. So the females that were in those warmer tanks, they also produced eggs in slightly lower egg energetic condition than the females in the colder tanks. So again, that tank temperature differential did seem to matter. Okay, so before we move on to part 2, I just want to quickly summarize what we've gone over and learned so far. So the first is that we found no effect on male energetic condition, but we did find that temperature effect in the female energetic condition. We also see that the CO2 level and the size of the fish both affect the males and females, but it does so oppositely. So for the females, we see a positive effect from ocean acidification and female size, and the inverse is true for the males. We also see this positive relationship between energetic status in the males and their reproductive output. And it's a little funky not to see it in the females. But one thing I'll kind of caveat with that is the time course of when we take these measurements is a little off for the females. We strip spawn them, continue on, and then dissect them a week later. The males, everything is collected in real time. So maybe it's a little bit of a time component. We saw nothing with the egg morphometrics, and then suspense. We do see effects on egg quality. So we do see a negative effect from the CO2 level and from temperature, but separately. And then we do see an effect of that egg concentration on the egg quality, but I wouldn't put the connotation that it's negative. I just think that it might be an inverse relationship or a trade-off that we see. Okay. So to summarize the summarize that we had, we do see variable responses to CO2, maybe extreme temperature sensitivity. And then for the males, we do see a negative effect of CO2 level, and maybe we see signs of sex competition, but we'd have to explore that further. All right, moving on to maternal effects, my favorite part. Okay, so for this one, we also have 4 broad categories. We're going to look at developmental success, survival, hatch timing, and hatch morphometrics. And like last time, we will always assess whether or not there's an effect of CO2 level. And then we will also look for any maternal effect. So again, that will include mom's energetic condition, something about her egg morphometrics, or egg quality. So to start off, for developmental success, what I didn't mention is when we fertilize those gametes, put them in the egg incubation beakers, we also put a subsample aside in different beakers, and we scored those 24 hours post-fertilization to look for developmental success. And we were looking for things like making sure the egg was fertilized, or there wasn't bad cell division or any sign of cell death. And in looking at developmental success, we actually find an effect of female energetic condition. We see that for the females in lower energetic status, they were more likely to have less successful egg development 24 hours post-fertilization, which suggests that there could be this influence of a maternal effect on offspring viability. Next, we're going to move on to survival. Survival is kind of a weird one because we were able to look at it at multiple time points chunked up and across the entire experiment. For our purposes, we're just going to look at larval hatching, which essentially means of all of the eggs that made it to hatch, how many of them actually hatched. And what that does is remove any weird things that might have happened during the egg incubation phase. What I will say is we have looked at all of it, and the results are all pretty similar. So looking at larval hatch success, we see an effect of egg morphometrics. Here we see that the eggs that were bigger actually had a lower success of hatching well, which is a little counter to what we typically expect to see in Arctic cod. The only thing that I can think of here is that we're looking at size in reference to diameter and not weight. And sometimes there can be some malformities that artificially kind of poof up an egg that would not be conducive to successive hatching. So we would have to dig into that a little bit more, but it was a little bit of a brain scratcher for us. And probably none of you care, but we were like, didn't expect to see that. All right, moving on to hatch timing. So for hatch timing, we can kind of look at this in 2 different ways. And again, that's because we have this prolonged hatch cycle that lasts about like a week to 2 weeks. So we can look at it categorically and characterize components of early, mid, and late hatch. But we can also take the cumulative hatch up to 100% and then look at it quantitatively at 5, 25, 50, and 75% hatch. And of course, they link up. So 50% hatch is going to be about mid hatch in that categorical view. So looking at hatch timing, we do see an effect of CO2 level. So just really quick on the bottom, we have that hatch cycle in terms of cumulative hatch, and then I just linked up the hatch categories. So essentially we see delayed hatch under ocean acidification conditions. That is more pronounced earlier in the hatch cycle, and then it becomes a bit more dampened as you get later in the hatch cycle, but it's still significant towards the end of hatch. So there was a pretty significant delay in hatch. Okay, and then finally looking at hatch morphometrics. So as I said, we were able to photograph the larvae, and that's so later we can look at things like the length, body depth, and yolk area. For the interest of time, we'll not look at the body depth results, but the length is useful because size at hatch can be a good indication potentially of how well larvae will do in the wild. And yolk area represents the amount of energy reserves the larvae has, and they kind of have this ticking time bomb to figure out how to eat before all that yolk is used up. Another thing I'll point out before we keep going is these metrics are really sensitive to the hatch cycle. So we typically see kind of premature looking fish early in the hatch cycle, and then they become a bit more developed as you get later in the hatch cycle. So all of our results will be in reference to those hatch categories, and you'll see what I mean in a little bit. Okay, so first looking at the size at hatch, here we find that there is an effect of CO2 level and a component of egg quality. Before we get on, I just want to point out those hatch categories. So we've got early, mid, and late. And as expected, we have smaller larvae if they hatch early and bigger larvae if they hatch later. Essentially, the late hatchers have had more time to develop an egg. We see a positive effect of moms that provision the eggs with more polar lipids tend to have larvae that hatch larger. And this is really cool to see because essentially the polar lipids, right, are these structural membrane lipids, which are kind of the building blocks to build that larvae inside of the egg. But also for gadids, which arcticod are a part of, they can also be an energy or fuel source that are also used in the process of embryogenesis. So for me, it was just kind of cool to see. And then we do see an effective CO2 level. The larvae hatch a little bit bigger under high CO2, but this actually might be a developmental signature because those larvae hatched a little bit later. So it might not be all CO2. And then looking at yolk area, we also see an effective CO2 level and again, egg quality. And we're paneled again, early, mid, late. And here, as expected, the yolk area is really big in those early hatchers, and it gets a little bit smaller as you go later in hatch, and that's because the late hatchers use up the yolk while they're still in the egg. But we see this really tight relationship between total lipid and yolk area. And if you're thinking to yourself, well, duh, what I want to emphasize is these metrics are measured really far apart in time. So we're looking at the egg that the mom provisioned inside of her ovary, and then we're looking at the amount of yolk available once they hatch, which is over a month later. And there's so many things that can happen between mom developing her eggs and the larvae hatching that can use up yolk. So there is this tight linkage, and that could be very beneficial to the larvae with big yolk. And then finally, we see this decrease in yolk area at hatch with high CO2. Again, though, that could be that developmental delay in hatch thing that we're seeing. Okay. So before we end, we'll just quickly summarize what we've seen so far. For developmental success, we see that tied to mom energetic condition, moms in lower energetic condition have lower developmental success. For the survival, we actually see this component of egg morphometrics, the egg diameter, influencing hatch success. Hatch timing was affected by CO2, it was delayed. And then for hatch morphometrics, we have a sort of flippy-floppy dynamic with CO2, but I think it's related to time. So I wouldn't read into that one too much, but then we do see that egg quality has an influence on both of our hatch morphometrics. And to summary the summary, we do see variable responses to high CO2, and we do see maternal effects. We see that mom's energetic condition can influence her offspring, and then also components of that egg energetic condition can affect the larval hatch morphometrics or the egg quality. Okay, so this is a summary of like tons of work. And I just want to end by thinking about what some of these mean in the wild, right? So far, we've been really like head in the lab, but so what? Why does it matter in the Arctic? So the first one that we'll look at is extreme temperature sensitivity. I just want to emphasize our tank temperature differential was minuscule. Again, 0.1 to 0.3 degrees daily. To put that into context, if we were to run a reproduction experiment, that tank temperature differential would be between maybe 2 to 5 degrees, and then have that add up over time. So it is pretty surprising to see. We do see that the females in those warmer tanks had lower energetic condition post-spawning, and we also saw that the eggs from the moms in the warmer tanks had lower egg energetic condition. So this suggests that they might really be affected by warming in terms of reproduction. We are actually following up on this, so stay tuned for that. But I just want to think about this a little bit more broadly, and that, remember how we said these are total spawners? And so what we might also be seeing here is that sensitivity is driven by their spawning strategy. They have that one giant draw of energy and that giant release of eggs, and that might make them more vulnerable than, say, a fish that has multiple times to sort of meter off eggs throughout a much longer spawning season. So just an idea. Okay. Now we're going to move on to that CO2 component. And you might have picked up that I didn't mention CO2 all the time. And in reality, where you saw an effect of ocean acidification is pretty much where we found it in the data. There were many, many metrics that were unaffected by ocean acidification. But we did see some responses. We actually found opposite responses between the spawning adults. We found a negative response in the male reproductive output, but a positive effect on the female reproductive output. But then we did find the signature of elevated free fatty acids in the female eggs from high CO2, and that could indicate maybe some lipid breakdown that's occurring. But regardless of some of these negative or positive effects that we're seeing, we didn't really see any effects on the success or the mortality rate of these individuals. And what's interesting about this is the only other study focused on Arctic cod hatching with ocean acidification found a negative response under ocean acidification. But those Arctic cod were also from more of a European population, and those adults were not held at high CO2. So maybe we are seeing some parental benefit from exposure. And then finally, we did see this pronounced delay in hatch timing under ocean acidification conditions. And the thing that I want to point out is we ran this experiment at 2 degrees Celsius, but in reality, these fish are likely closer to 0 degrees or those eggs underneath the ice. So that delay in hatch would be even more exacerbated underneath the ice. And so we kind of have this two-parter thinking about ocean acidification. We might be seeing some benefits from parental exposure. You'll notice that we didn't do across, and I can talk about why we didn't do that if anyone is curious. And then we also found this delay in hatching. And in the wild, this is concerning because it could mean that there are the sort of cards set up for a prey mismatch type of dynamic where the larvae hatch and it's not well timed with the available food. So something to think about for them. And then finally, we did find some maternal effects. So we see that the moms in lower energetic status were more likely to have less successful egg development. And we also found from the eggs from the moms that their egg quality also mattered. So for the moms that provisioned the eggs with more polar lipids, those larvae hatched a bit bigger. And for the eggs that had more total lipids, they also had more yolk. And in the wild, being bigger and having more yolk really could set up an advantage to surviving your first larval season than being a really tiny larvae with not a lot of yolk. So what that means is maternal condition and egg quality could both indirectly or directly and indirectly affect offspring survival. Okay, so with that, I just want to sort of emphasize again, broad for our experiment. We found maybe some extreme temperature sensitivity, but we're following up on that. But it was a surprise to us. We see these variable responses to CO2, and it might matter in some metrics and it might not in others. And then we did find maternal effects, which for like the nerd in me, that was really exciting. And with that, I am happy to take questions.
That was a lot of data. Trying to get my head around some of that. So I think we'll open it up for questions. We'll start with questions online. Roseanne, any questions? Okay, for anybody online, go ahead and put those questions into the chat box and then we'll work through it. Questions in the room. Anybody? All right, hang on one second.
Thank you for an amazing talk, Emily. I was curious, the temperature sensitivity seems crazy that you said it's so few fractions of a degree that cause the effect. So I was wondering if maybe you could give us a teaser of what the temperature follow-up experiment you're going to do is.
Yeah. One thing I will also add is In the spectrum of responses that you could have to environmental stressor, seeing like a slight effect is interesting to see in terms of temperature, but it also didn't lead to any like over mortality or other things like that. So just to put that into perspective, but then actually Luis is heading it up. We've got young Arctic cod who will be, if they do spawn this year, that'll be their first time. And they are in temperatures of 0, 2, 5, and 9. They're cooking. And then they also are in 2 different food treatments as well. So a high and a low. And then tracking reproduction. And then Luis does a bunch of the lipids. So focusing on a lipid component too. Yeah. That was for the fall period. Now for the winter. Yeah.
We're good? All right. Roseanne has a question online I can see.
Can you talk about how you determine egg quality and how you determine the lipids and lipid quantity in them?
Yeah, so egg quality I used as a umbrella term for these lipid dynamic components. So all of egg quality is related to lipids. And then in terms of quantifying the total lipids, Luis has a wonderful lipid lab in our NOAA facility. And so we're able to extract the lipids using a Folch procedure with chloroform and methanol. And then we just get the amount of lipid from that procedure and then can take that lipid and further look at the lipid classes in a technical way that I don't know how to, I didn't run the lipids. So, but in magic and with gases and chemicals, if you have any follow-ups, just let me know. Thin layer chromatography.
Perfect. All right, question in the room.
Thanks, Emily. That was very engaging. Let's see. I have sort of related questions on the delay in hatch timing. I'm curious, well, what your thoughts about what's going on. I take it you measured oxygen and kept it high. And then, yes, I'm interested in your thoughts on the switching. I forgot the term, but switching them in from the treatment to the effect.
Yes, yeah. Okay, so for the delay in hatch, I don't know. But I think, so some of the reasons that eggs, so okay, let's back up. We would likely have expected that they would hatch early. The idea is that if there's some metabolic constraint in the egg, it is time to hatch and your oxygen delivery is a lot better once you are a nice larvae with your permeable skin soaking up all of the oxygen. And so under stressors, we typically expect slightly earlier hatch. So what could lead to a delay if it's not some sort of energy component? One species has essentially there is a specific enzyme that helps break down the chorion, which is how the fish gets out of the egg. And that enzyme could be pH sensitive. So there could be a dynamic there, or there could be other crazy feedbacks that were kind of just cross-wired under ocean acidification. But I would have to think about that one a little bit more. And then in terms of why we didn't cross, so the pragmatic reason is because we have to keep track of mom. We had an inkling through many other studies that there are maternal effects. So we wanted to keep track of the batches of eggs from the moms. We had to replicate that. And then we had different tanks and then different treatments. And so replication-wise, it would have just absolutely blown up. But in reality too, it is unlikely that a switch like that would happen in the wild. But I would have loved to do a cross just so we could maybe knock out what really was parental benefit. But there are other tools that we could use in the future related to like genetics or gene expression or other things. So yeah, it'd be worth revisiting.
Okay, I'm going to ask a question. I'm just going to be selfish. In that delayed hatch, did that change the overall duration of hatch time? So first day of hatch to very last day of hatch to that batch, or was it just a delay in the first hatch?
It was funky. It didn't significantly affect the hatch duration. So they were all kind of like shifted. but it was more, if I were to like plot the density, essentially from the end to end, it didn't change much, but we did see a shift in the synchrony. So the high CO2 ones that were delayed, they had a tighter synchrony of hatch. And then the ones in ambient had a bit more of like a nice kind of curve out. So yeah, I don't know if that answered that.
Yeah, that's
okay.
exactly what I wanted to know. Hang on just a second. Question online at all, Roseanne? Okay.
Following up on that, the original graph you showed where you had on the x-axis the percentage that had hatched, and on the y-axis the time, you only went out to 75%, and it looked as though if you took that out to 100%, they would meet. Then on your summary slide, you took the hatching and simply moved it, which doesn't match that data. My question is, I guess it's a different way of asking what Cinnamon asked. Did they all hatch, and is there an effect on the number of eggs, the overall number of eggs that hatch that is linked to CO2?
Yeah, good question. So I'm actually, okay. So in this graph, so kind of why there's also a 5% is we typically see this early blip. So it's not like 1 or 2, it's like a little pulse of really early hatchers that happens. And so we found that this blip was the most sensitive to CO2 level. That was the most different in terms of delay. And then as we get into more and more fish across the mid time, then that's when they start to converge. And then I end at 75 because beyond that, you kind of start getting just weird stragglers. So yeah, to answer your question, and I could go back into it more, when I just calculate the length of total hatching, the synchron, and this, or there is no change with CO2 in the total length. We do see a slight shift in the synchrony, which means that the length is not being affected by stragglers that persist in the high CO2, either the ones that did go a little bit early or went a little bit late. So yeah. There's also the problem too is the sample sizes are not huge and there's a lot of variation. But in terms of my clip art, that was artistic interpretation and should not be taken as a journal ready figure. But for the purposes of this, yeah, I don't know, the timing is, I think what's confusing with the timing is there is a lot of variation. And so it's trying to figure out how to group things together in a meaningful way to analyze it, also as it's across a full spectrum. But we do, like, for the cumulative hatch proportion or percentages, that comes from daily data.
Great, thank you. Other questions in the room? All right, second.
Oh, and it wasn't affected to survival. No.
Hi, great talk. Question for future studies. Is there anyone working on it, or do you have any plans of working on just seeing the development over time? So instead of just ending it after hatch, continuing on to like maybe trying for first settlement or going into like first spawn and just kind of doing a long-term analysis of the CO2 on them?
Yeah, that would be lovely. Yeah, we actually had these talks about when to end it. And we did think about what if we even just ran the larvae out to once they use up all their yolk and not feed them. But in reality, after almost 6 months, we were done. Also with that, so in order to get all these beautiful things about hatch, we had to sacrifice all the individuals at that time point because we're taking out the hatchers as they happen. So there would have also needed to be the additional space to have more babies that we don't touch that we carry out to like, yeah. So I think that would be the pipe dream. And maybe nowadays less likely, but definitely something we could think about in the future. Yeah. Also with a big problem too is with the adult component, we crank through the CO2 bottles, and so it's really expensive. So we also need to like, yeah.
All right. Looks like we got one more question. We got a question behind me first, which I never see. I'm sorry, Joe.
Just a follow-up question related to comments about what egg synchrony can mean in the environment. Since everything was strip spawned, and I assume at the same time, could there also have been an influence on maturity timing, which could either have exacerbated that asynchrony or wiped it apart?
Yeah, good question. So actually they will not release eggs unless they're totally ready. So the accumulation of fertilized gametes happened over about 2 weeks. So not all the moms were synchronized. And we did look at if the date of when the mom was strip spawned had an influence on anything, and it didn't seem to affect much. And they are all the same age. So they're also in that component. But yeah, but we could look more into the timing of the different females. Yeah, does that answer your question? Yeah. No, because if they're not ready, nothing will come out. So like we would kind of test, and then it's either like she's not ready, or it's like get the beaker. So which was all, well, she's gone now, but Mary Beth was the squeezer.
Amazing talk. Just really so much data and so well presented. One of the questions that I had was one of your other graphs where, and something, follow up on something you said, there was so much variability. I looked at the data points on the graphs, and then it looked like you had lines, right, which were linear, but you also appeared to have them bounded. Were those 95% confidence intervals or what? I was just wondering if that was, if I was interpreting that right or that.
So like one of these?
Yeah.
Yes. So this is, so yeah, so these are all the raw data, and then these are the model fit from the best fit model. And then for these, it's actually 2 times the standard error from the model, or the standard deviation from the model.
Okay.
So like,
yeah,
similar,
thanks.
yeah. And then the ones that were kind of curvy, those are because we were looking at proportional data, so it's all beta regression, and we can include a beta parameterization component if that's a better model fit. So that's why some of them had the curve.
Thank you.
Yeah.
All right, last call for questions. Oh, got lots more questions. Hang on.
So thank you for this really great talk. I really enjoyed it. At the very beginning, you showed a graph on the energetic costs. Yeah, that if there's a stressor, then the maintenance is increasing, and there's less energy for reproduction and growth and the other parameters. So my question would be, did you measure any metabolic rate or something?
We did not, and I would have loved to do that. Yeah. Yeah, I think if we were to add another physiological component, it would definitely be in the females, kind of see what's going on with them. Because it is funky to see an increase in fecundity, and one of the running hypotheses is there's some interaction with a change in internal pH and how that changes hormones. And so it could act as like a stimulus. And these females are well fed. So it's like if you could stimulate a little bit more production, they might actually have the capacity to increase that production. That is kind of like one of the ideas for the increased fecundity, but it is kind of puzzling.
But did you look, did they eat more? I mean, under ocean acidification or something?
We didn't do directed feeding studies, but in just our normal husbandry, it didn't seem that they ate more or less than other ones did. Yeah. But For the feeding, we kind of did our normal routine feeding. So usually we just make sure that there's not any tanks that are having a lot of additional scraps of food. That would indicate a slowdown of feeding. So we didn't see any of that. Sometimes it a little bit once they're like just about to spawn. But yeah.
Yeah,
We can also talk more. It'd be fun to think of some follow-ups. Yeah.
Great. I think that we're going to let you off the hook. So one more round of applause for Emily, please. And while we wrap up, will you go to your last slide where your contact information's at?
Oh, yeah.
So then if you have additional questions, feel free to reach out directly and we'll get those answered. Thank you very much. And for everybody here in the room, I hope to see you again next week. And for folks online, thanks for being here, everyone. Thank you.